much, Bernadette, for a very kind introduction, and thank you to Fran and the other organisers for the invitation to give this Paget lecture. It really is a huge pleasure and honour to do it in the context of these inspiring awardees, and also at the end of a list of really alarmingly august predecessors. So thank you very much for the invitation. So the theme of the lecture, as you've heard, is ageing. And I think ever since people have started writing down their ideas, there have been notions of immortality, of being able to turn back the clock of time on ageing, pushing the old Rembrandt back to the young one and having perpetual youth. And just down the road at the Royal Society, I think one of the first scientific statements of this came from Robert Boyle, because he made a wish list of the things that he thought that science should solve, the most important scientific problems, but the ones that were probably going to be the hardest to crack. And the two top items on his list were the prolongation of life and the recovery of youth. And he didn't live long enough to see it, but the prolongation of life is something that's been happening very steadily in developed countries since the middle of the 19th century. And it's illustrated here by the work of two demographers who've plotted, against the year in which the births took place, life expectancy for the country that was the world leader at the time. So to start with, this was the Scandinavian countries, went through various others in the middle. It's the Far East now. I think Hong Kong for women and Japan for men, just at the moment. And you can see this astonishing steady increase. It's gone at about 2.5 years per decade. And the horizontal lines here simply represent various individuals and organizations' prediction of where the trend would top out, what the intrinsic limit on human lifespan would turn out to be. And you can see it shot past all of them. This was the basic point of this article, and it continues to be true. There's no clear signature in the demographic data of what the limit will be, although in practice, this trend, at least in some countries, may turn back with the tide of obesity and metabolic disease amongst the young particularly. But I think it's a trend to be celebrated because of improvements in civilization, in healthcare, different things at different points in this career, so this trend, immunization, antibiotics. Towards the end, the increases in lifespan are happening almost entirely in the elderly part of the population. And as a result of these things, people are staying healthier as they age and living longer. The world record holder, some of you may well be familiar with her, a French woman, Jeanne Calment. She died at the age of 122 and about a half. She actually met Van Gogh in her father's shop when she was young. He used to go there to buy paint. It's interesting, I think, to speculate about her, what her secret was. She came from a very long-lived family, and human lifespan is about 25% heritable, although that heritability is concentrated in particular families. On the other hand, she wasn't a terribly good advertisement for a healthy lifestyle, which we might expect to see. She actually gave up smoking when she was 119. So who knows what her secret was. But that's the positive side. Of course, the aging, the increase in lifespan is coming with some downsides, some of them temporary, some of them harder to crack. One is this one, simply the change in age structure that's happening in most developed countries. So what we've got here is a stack of the different age classes, the oldest at the top, the youngest at the bottom, for 1901 on the left and 2010 on the right. This is just for the British population. And you see the Christmas tree shape gradually going apple-shaped. And this is very typical for most developed countries, including European countries. I noticed the Pope the other day described us as elderly and haggard in Europe. And this brings with it certain economic problems, particularly to do with pensions, but also intergenerational conflict over wealth and so on. But for this evening's topic, which is the science of aging, the problem is this one, which is that increasing age is the major risk factor for the chronic and killer diseases. So dementia, cardiovascular disease, and cancer. These are just statistics from various UK and European sources showing the trend with age for the two sexes. Cardiovascular disease, there's been considerable progress with, and quite a bit with cancer. The one that's proving really intractable at the moment is dementia, and it's both very prevalent and very expensive. So these were just some figures forecast a few years ago now. The trend hasn't changed since then for what the prevalence of dementia in the UK will be by 2051. It's about 1.7 million people. And that, not just for Britain, but for developed countries generally, is absolutely enormous, and a significant proportion of the national budget potentially going on care. And there have been a number of failed clinical trials for Alzheimer's very recently, so it really is proving a hard one to solve. And interestingly, and I know there are some funders here who might be interested in this, research into dementia is really relatively underfunded. If you look at the cost per patient per year of treating, for dementia it's much higher than for the other three, cancer, stroke or heart disease. But if you look at the funding, it's way down. So relatively, I think we're not putting enough into this problem. But coming on to the science, given that increasing age is the major risk factor for these conditions, one obvious way to set about potentially trying to tackle them might be to intervene in the underlying aging process itself, because it's the common risk factor for all of them. If we could intervene in that process, then perhaps we could get rid of the major risk factor for all of them simultaneously and push back their incidence or ameliorate it. But until recently, the prospects for doing that have not looked particularly rosy. One reason is that aging is a very complex process. So this is simply a picture of what was actually originally a table produced by a pathologist called George Martin. And he listed the different human tissues and the things that can go wrong in them during the aging process. And what you see is a picture of many different things happening within a single tissue and quite different things in other tissues. It looks as though what we have here is each tissue in parallel encountering different kinds of insults of daily living and developing damage and pathology accordingly. There is no single aging process here. It looks unlikely that a simple intervention like a single gene mutation or a drug would be capable of capturing all of these or even a lot of them simultaneously and slowing them down. It looks like a case for, at best, piecemeal intervention into different problems. And by the same token, I think people have often thought that a similar argument would apply to these creatures, the laboratory model organisms, because they're, of course, the real workhorses of modern biomedicine. Often, if we want to understand a process, we start simple, either with a bacterium or a single-celled eukaryote, like the yeast up at the top left. Once we've worked it out there, we go and look at the multicellular invertebrates, the worm and the fly, and only then go on to the more complex mammalian system. And that whole process works because of the astonishing evolutionary conservation of many biological processes for genetic transmission, expression of genes, metabolism. Many things are so conserved that we can take a gene from one of these organisms, or even from a human, and put it into another, and it works just fine in the new context. The function is that conserved. But it seems perhaps a little intuitively unlikely that that's going to be true for aging. For a start, these organisms have very different lifespans. The worm's about 3 weeks, the fly's about 3 months, the mouse is about 3 years. They have very different diets. They undergo different stresses in the conditions in which they live. Their life histories are very different. 3 of them are cold-blooded. Why should they encounter the same kinds of insults of daily living? Why should their problems as they age be the same? And the reasons that aging research has become really exciting in recent years is because of the realization that, at least to some extent, those pessimistic ways of looking at the aging process are wrong. And the aging process does turn out to be malleable. It was first discovered through the work of the person who's my personal hero in this whole subject area, who was called Michael Klass. And he did a very simple experiment with the worm. What he did was to ask whether, if he mutated, just randomly mutated the genome of the worms, whether he could recover long-lived strains. So can a mutation in a single gene make the whole worm live longer? And he found that he could. And this is an example of one of these mutations. Actually, this is more modern data collected by one of my colleagues at UCL, David Jams. And what we have here is a single gene mutation. I'll explain what it is in a minute. with the control survival curve in blue and the mutant in red. And you can see that there's an absolutely enormous increase in both the median and the maximum lifespan of the mutant animal. And very interestingly, this effect turned out to be evolutionarily conserved, because what we have here is a mutation in the single worm insulin-like growth factor receptor, a signaling pathway whose existence in the worm was not suspected actually until this gene was cloned and sequenced. What we're looking at here is a set of signaling pathways much more familiar in the context of control of growth during development and in control of metabolism, but now turning out, at least in the worm, to be important in determining its lifespan. And this effect of insulin-IGF signaling turned out to be conserved first to Drosophila and then to the mouse. So these are just some of the mutants here. You see the fly in the middle. This is a mutation in a gene called Chico, which encodes a protein which just sits just inside the receptor in the cell. And you can see the nice extension of lifespan in the flies that have lost either one copy in orange or both copies of the Chico gene in red. Quite a big extension of lifespan. And on the right, we have a Chico mouse. So this is from the lab of my colleague Dominic Withers, involved in a Wellcome consortium working on these mutations. And you could see that in this female mouse who's lost both copies of this Chico equivalent, again, we see this nice increase in lifespan. One of the odd things about the pathway is that when we mutate it, generally it extends lifespan more in females than in males. We don't know why that is at the moment, but it's quite a consistent feature. But very importantly, although lifespan's a nice readout, it's the sort of gold standard for whether you've actually managed to slow down the aging process, it's also quite a blunt instrument, because really the aim of this research is to try and improve function and health during aging. So there's been a lot of work on these mutants, looking at whether what we're seeing here is simply an extension of the moribund period at the end of life or an improvement in health. And for the two invertebrates, there are a number of readouts one can use, usually based on mobility, but also learning ability, intactness and function of individual tissues, and so on. And these mutations do improve the health and function. Very importantly, in the mouse, this particular mutant was quite thoroughly phenotyped. And what they found was the sort of broad spectrum improvement in health that we hadn't been expecting to see. So these mice, as they age, they actually start out life slightly insulin resistant when they're young. They look as though they might get diabetes when they get older. But in fact, as they go through middle and old age, they cross over with the controls. And later on, their glucose handling and insulin sensitivity is very much greater. They've got a better immune profile, more naive T cells. They maintain their motor performance better. They can hang on to a rotating rod better as they age. And they also show a delayed onset of some specific aging-related pathologies that these mice get. So they get less osteoporosis, less cataract. You can see on the control mouse there, the right eye, as we look at it, has got a cataract. And also you can see this ulcerative dermatitis on the head and the nape. About 40% of the control strain get that at some point during aging. The mutant mice are completely free of it. We've never seen a case. So these systems don't obviously have a connection with each other, yet they're all improved by this one lesion in a single gene. And I think it's this sort of finding that's making people sit up and pay attention. Of course, what's important here is this going to be the case in humans? And there are a number of studies of aging human populations, looking both at their physiological and other characteristics, but also at their genetics. And the way that that's usually approached, because of limitations on sample size, is to look in the humans at the genes that are the equivalents of the ones that have turned out to be important in the animal studies, and to look at whether particular variants of those genes are associated with survival to late ages. And these are just some of the results from those kinds of studies. So you'll see on the left here, this FOXO comes up a lot. FOXO is a particular type of transcription factor that turns expression of genes on and off, and they're key players in this insulin-like growth factor signaling pathway. And it turns out that in humans, key genetic variants, and 2 of them, are associated with survival to 90, 100, or even beyond 100. And so also are variants in the insulin-like growth factor receptor. That's also turned out to be an important player. So it's early days, but the genetics is quite encouraging, and particularly the physiological characteristics of these survivors really do look as though the insulin, IGF, and also the associated target of rapamycin pathway are very important in determining the health of people as they get older. So what we've got here, as I've said, is a signalling network that normally senses the nutrient status of the animal, the presence of growth factors, certain stresses that it might be subject to, and then decides how much of metabolically expensive processes can be afforded. So how much growth, how much reproduction, what type of metabolism, and now turning out to be important in lifespan. And almost certainly what we've tapped into here is the physiological mechanisms that bring about the response to an environmental intervention that's been known about for a lot longer, which is dietary restriction. So it was first discovered in rats way back in the 1930s that if you put the animal on a diet, so you force it to eat, say, 60% of what the rat would like to eat if it was left to choose for itself, you see a big increase in lifespan and a very broad spectrum improvement in health during aging. That's also turned out to be true in mice. And then subsequently, in all of these both model and highly non-model organisms, it seems to be a very evolutionarily widespread response. And recently, there have been a couple of studies in the United States on rhesus monkeys, very long-term studies because they live for up to 40 years, so pretty heroic work to do it. But just some of the output from the first one to be published is on the left here. And in this study, the Wisconsin study, they found both an increase in lifespan of the restricted monkeys. They took them down to about 70% of the intake of the controls, and a huge protection against various cancers, diabetes. There was virtually no diabetes in the dietary restricted monkeys, improved cardiovascular health, and also improved cognitive performance. So again, this broad spectrum effect. The second study actually didn't find an increase in lifespan, but it did find the health indices improvement, very similar to the first study. And the differences between the two, I think, could be explained quite easily by details of the way that they were done, if anyone wants to discuss it later. In humans, there isn't a huge amount of data, the main reason being that most humans don't have the self-discipline to subject themselves to dietary restriction. It's an extremely difficult thing to do, to actually eat little enough to get the health benefits. But there are a few individuals who are prepared to do it, mostly men, interestingly. There's quite a sex bias there. And this was just an example of one study where they looked at voluntary restrictors and looked at them once they'd been doing it for about 7 years versus controls who were matched for various characteristics. And in this particular study, there was a big improvement in 8 different markers that are risk factors for atherosclerosis. So we don't know with humans really yet how effective this intervention is going to be. There's a very big long-term study just set up in the United States called Calorie. They have a web page and they've just recruited their cohort. So I think it's going to be very interesting to see the outcome of that one. So that's just a very brief account of the developments in the research area over recent years. And what I'd like to do for the next bit of the talk is just talk a little bit about dementia and the nervous system and the way that some of these interventions into aging may give us some hope that there could be ways of preventing dementia and specifically Alzheimer's disease. So I do a lot of my work with the fruit fly, Drosophila, also with mice. But the work I'm going to tell you about that we've been doing is mainly with the fly, and then I'll include some mouse work as well. So what we wanted to do was to develop a fly model of the normal form of Alzheimer's disease. So as I'm sure most of you are very well aware, it's the most common form of dementia, and at the histological level, if you look at the brain of patients, then there are 2 very obvious markers of the disease. One is these amyloid plaques, which are made of a little peptide called A-beta, and the other is neurofibrillary tangles, which are inside the cells. The plaques are outside, and the tangles are made of a protein called tau. there's early onset Alzheimer's disease, which has a clear family pedigree associated with it. But the vast majority of cases are actually sporadic and with no previous obvious risk factors, apart from aging, which is the major risk factor for the disease. So what we wanted to do in the fly, and these are the people who did it, 3 postdocs and a grad student called Ian Rogers, was to develop a fly model of this sporadic late onset form of the disease. So what we decided to do was to produce a fly that goes through its development completely normally. So it comes through to adulthood fine. But then we arranged to turn on in the nerve cells of the fly this peptide, this amyloid beta, very short peptide, that's associated with the disease in humans. So we actually use the human peptide. So the fly has an equivalent of the protein that in humans gives rise to the A beta fragment by cleavage. But if you cleave the fly protein, that bit's not conserved and it's not toxic. So what we're doing is introducing a new, potentially toxic protein into the brain of the fly. And to get it expressed only in the nerves and only in the adult, we use a system for gene misexpression in the fly, which is actually derived from yeast. It's called the Gal4-UAS system. And what you have is a Gal4 driver, which drives the expression of the gene that you're interested in, and you can put a promoter in front of it, in this case LOV, which is expressed only in nerve cells. And you then cross that fly with one which is carrying the upstream activating sequence, which is driven by the Gal4, and then expresses the A beta toxic peptide. But it's only expressed in the presence of an inducer. If we put these two together in the progeny and just leave them, nothing happens. So they can come through development normally. But we then add to the food the inducer, which is a steroid hormone, a drug called RU486. It's actually the active constituent of the morning after pill. And if you add that, now the Gal4 really binds to the UAS and drives expression. So what we can do is to confine expression to the adult neurons of the fly. So we did that with a known toxic form of A beta and had a look at what happened to the fly. And the first thing was that there was quite clear evidence of neurodegeneration. So this is shown here. What you're seeing here is the mosaic appearance of the eye of the fly. This is made up of lots of parallel photoreceptors. Here is a photoreceptor. And what you can see at the bottom is that one of the photoreceptors, the one in the middle, has actually been lost in that fly, whereas they're all present. There should be 7 in the top fly. And as the flies get older, we've got 2 controls here. On the left, we've got just the driver to make sure that it's not doing anything naughty to the eye of the fly. We've got a non-toxic form of A beta, and then this toxic one that we're interested in. And you can see that as the flies get older, what we see is a progressive loss of the photoreceptors when the toxic protein is present. And in fact, by day 25, they're all dead, so we couldn't measure it. So there's clear neurodegeneration in the flies. We can also show that there's a decline in the function of the nervous system using electrophysiology. So to do that, we can use this particular part of the nervous system in the fly, which mediates the escape of the response to a frightening visual stimulus. So it's perceived at the front end, and then goes back through those 2 giant interneurons in red, and either synapses directly with the motor neuron to drive the trochanteral muscle, which depresses the leg so the fly jumps, or indirectly through an interneuron with the motor neuron that innervates the dorsal-longitudinal muscle, which is the flight muscle of the fly, so it then flies away. And this is a simple and very well characterized system. We know what the synapses are made of and the exact anatomy, and we can put electrodes into the eye of the fly and record from the muscles. So we can stimulate through the eye and then ask in the muscles how well is the electrical impulse being transmitted through this system to affect the escape response. And what we found was that in these Alzheimer's flies, it's impaired as the flies get old. So you can look simply at how long it takes the impulse to get there, the latency, and you can see that in the old flies here, the red Alzheimer's flies are taking longer. You can also give the fly a repetitive train of pulses and see how many of them are picked up by the muscle, which is a measure of how quickly the system recovers from a previous impulse and can respond again. And again, very obviously, as the flies get older, you start to see an impairment in the flies expressing the toxic protein. So definitely an impairment of function, and we know actually that this is to do with impairment of synaptic function in the system. We can also do a behavioural output with the fly. One of the things about flies is that they like moving upwards. Generally, you usually find them at the top of the vial. So you can measure the extent to which they're capable of exerting that preference by simply banging them to the bottom of the vial and looking how long they take to get to the top. And this is shown here for the controls on the left and the mutant AD flies at the right. And you can see that the wild types are coming up nicely, whereas although they're moving, the Alzheimer's flies are stuck at the bottom. So there's a behavioural impairment. This is just the quantification of that. So it's a response that also goes down with ageing. Older flies don't respond so readily. And you can see that the age-related decline is much sharper in the Alzheimer's flies. And also, they show a decline in lifespan. So I think here we have a quite reasonable fly model of many of the things that are known to go on in the nervous system in mammals during aging. And we used it to ask two very simple questions. One of which is, why is aging a risk factor for Alzheimer's disease? What is it that's actually going wrong in the neurons of the fly when this protein is present? And why is it worse when they're older? And the second was, can we rescue any of these phenotypes that I've told you about with altered insulin IGF signaling, the kinds of mutants that also extend the lifespan of the fly? So as far as why is aging a risk factor goes, we can look at this at the molecular level by asking, first of all, how does the fly handle the toxic protein? Is there a problem with getting rid of it from the cells? And we could also ask, suppose that a fixed amount of the protein is present, do the aging neurons respond worse to the presence of that peptide? So those were the two experiments that we did with this setup. So again, as I'm sure almost everybody knows, there are two stages in gene expression. When a gene is expressed to produce a protein, there's an intermediate molecule called RNA, and then eventually that goes on through translation to produce the protein. And in this system, what we can do is arrange to give the flies a pulse of the RU486 inducer that produces exactly the same level of the RNA in flies that are induced when they're young, only 5 days old, or 20 days old. And the minute we remove the inducer, almost within hours, all of the RNA disappears. So the intermediate molecule is a very acute response to the presence of the inducer. And we can look at what then happens to the A-beta peptide in these neurons. And I think what's interesting here, so this is the flies after the inducer has been removed, so there's no further RNA present. And you can see that in the young flies, there's a much lower level of the protein. Neither of them manages to clear this protein. It's insoluble after the inducer has been removed. It stays the same for the rest of their lives. But its level is much higher in the flies that were first induced when they're old. So it's very clear that for some reason, they can't clear this protein when it's produced, even though presumably it was being produced at the same level. And we think that this is to do with a decline in the activity of the proteasome in the nerve cells. It very clearly does go down with the age of the flies. But the result is that this brief pulse of induction at different ages actually results in a major reduction in lifespan in the flies that were induced later. So it catches up with them later on. So that's one reason the nerve cells have a problem. They can't clear the protein if it's present. But it also turns out that they have a problem with the protein itself, even if it's present at the same level as in a young fly. And this is shown here. So again, what we did was to titrate the system with the dose of RU486 that we gave them to produce flies that had got the same level of that insoluble protein left after the RNA had disappeared, with induction at 5, 20 or 30 days of age. So we're asking, is it worse for you to have this stuff present entirely in the old part of your lifespan? We looked at what happened to them subsequently. And you can see that on the right, the later it's induced, the worse off they are with respect to controls of the same age. So it's having a much more toxic effect on the neurons of the older flies. So there seem to be two different things that go wrong. We don't yet know the reason for the second effect. We're doing a lot of work on this at the moment. But one of the nice things about the flies, of course, is that you can get through these experiments relatively quickly. So there seem to be clear molecular reasons for why things go wrong with age. The second thing we've been looking at is this potential rescue of the Alzheimer's disease pathology with insulin pathway mutants. And we've taken a number of different approaches to this with different mutants. I'll just tell you about one of them because I think it's particularly relevant actually to the human condition and therefore of the greatest interest. And this involves a protein called glycogen synthase kinase 3, GSK3, much studied, and is very heavily implicated in the etiology of Alzheimer's disease in humans, although in quite a complicated way that doesn't always go in the same direction. So it's a key player in the insulin pathway itself. It's involved in the storage of glycogen, hence its name. It's increased in the blood cells of Alzheimer's disease patients. It's present at higher concentrations. In mammals, it's required for the generation of this A-beta peptide from the precursor protein that gives rise to it. Its level is regulated by the A-beta peptide, so there's a mutual interaction between them, and the molecular mechanism by which that happens is very well understood. It phosphorylates that tau protein, which is the one that makes the neurofibrillary tangles inside the cells of Alzheimer's patients. And there's a precise fly ortholog of the human protein. It's very conserved. It's actually called shaggy for reasons of the phenotype that it produces when it's mutated. Mutations of fly genes often have very capricious kinds of names. So we were interested in the potential interaction between GSK3 and our Alzheimer's disease fly model. And what we found was that actually if we produced mutations in GSK3, and particularly if we overexpressed it, that we could indeed rescue some of the phenotypes. So this is shown here for the climbing phenotype. I showed you that decline in the ability of the flies to climb with age and the fact that it was further impaired, and you can see that again here in red, with the presence of the A-beta peptide. What's shown in green is what happens actually if we overexpress GSK3. And you can see that there's a complete rescue of the climbing phenotype. So the life of the flies, that intervention is very highly beneficial. It also somewhat rescues the reduced lifespan, not completely. It's not as complete as this rescue. So there's clear age specificity of the effects, and also there's a yin and yang with misexpressing GSK3 because its effects tend to go in opposite ways in the nervous system and in the rest of the fly. So it's not a simple situation, but it's very clear that its overexpression here in the nervous system can rescue the presence of that peptide. And again, the downstream mechanisms involved in doing that is something that we're pursuing heavily at the moment. But interestingly, and I think somewhat in the other direction, we also managed to get a pharmacological rescue of this one, actually with treatment with lithium, which of course actually suppresses the activity of GSK3 beta. And we think this is going through a completely different route. It's actually at a dose of lithium that has very little effect on GSK3. And what lithium seems to be doing is increasing some of the stress response pathways in the nerves, in the nerve cells of the flies, particularly the cap and collar transcription factor. So there are a number of different things going on here which are providing a way in to interfering with the toxic effect of this protein in the nerve cells of the fly. And I think intervening in these aging pathways may well be a powerful way in here. And interestingly, these kinds of effects are conserved in mice. So I'm going to show you the work from a colleague of mine, Andy Dillon, who was working at the Salk Institute when he did this work. He's actually since moved to Berkeley. And what he did was to ask whether one of the mutations that, somewhat controversially actually, in the mouse extends lifespan, could also rescue the pathology associated with a mouse model of Alzheimer's disease. So the basic approach he took, shown here, he got a long-lived IGF-1 receptor mutant mouse. It's actually a heterozygous mouse, which he crossed with a double mutant Alzheimer's disease mouse. So it's a mutation both in the Alzheimer's precursor protein and in one of the proteins, one of the presenilins that cleaves it. So these are two different mutations that in humans are associated with familial Alzheimer's disease. And from that cross, of course, he can generate 4 different genotypes, and the one he's particularly interested in is the one on the right, which is the rescue of the pathology of the Alzheimer's disease on its own. And they took a number of approaches to looking at this, but one was this classic test of cognitive ability in mice, the Morris-Water maze. So the way that this test works is that what we have is a platform there, which I hope you can see with the pointer. It's just hidden under the surface of the cloudy water. And the way that the mouse is trained initially is with the platform above the surface of the water. The mouse is then introduced to the murky water and swims. It can see the platform, so it swims straight towards it. So it gets the idea that if it swims, it's going to be able to get itself out of having to swim. Actually, a minority of mice roll over on their backs and float and don't play the game, but most of them do. So what you do then is to take the platform below the surface of the water so the mouse can no longer see it. But you can see these landmarks on the wall at the back of the maze, and the mouse can use those to learn the location of the platform, which it's also had an opportunity to see previously. So what happens with an Alzheimer's, that double mutant Alzheimer's mouse, is that they swim well enough at this age, although that becomes impaired progressively later on. But they don't have a clue where the platform is. They don't even search in the right quadrant. They're just all over the place. Whereas, if we look at one of the rescued mice here, he largely confines his search to the right bit of the maze and actually quite quickly finds the platform. There he goes. The other one, as you can see, is still hopelessly swimming around. And if you quantify that, so this is the rate at which the mice learn, we've got the three control genotypes, or the two control genotypes in the rescue down below, and the Alzheimer's mouse at the top in red. And you can see that there's a very significant impairment of their ability to learn the platform. So this IGF-1 mutant's affecting a rescue of this learning problem. And also, to some extent, rescuing these other features of this mouse model. So there's, as I said, they gradually do lose their motor coordination, and the mutant somewhat rescues that. You can see the neuroinflammation here. This is astrogliosis in histological sections of the brain of the mouse. You can see how marked that is in the Alzheimer's disease, the second from the right, the rescues to the right of it. So it's not a full rescue. They're not as good as the two controls, but a lot better than the mutant. And it also rescues the actual loss of neurons in the model. So the mechanisms for that one are not understood at all. It's very slow work trying to go downstream in the mouse, but it's clear that similar kinds of phenomena to the ones that we can analyse more quickly in the flies and the worm may well be going on. It's not a simple one-to-one mutants that extend lifespan rescue pathology in these kinds of models. Some of the mutants that effect a rescue, for instance, a mutation in IRS2, the second insulin receptor substrate in the mouse, very clearly don't extend lifespan. Those mice actually get diabetes and they die young. But nonetheless, those mutants in the nervous system can sometimes rescue. So we really do need to understand what the rules are to figure out what's going on here. But clearly it's something interesting. So, of course, what we would like to be able to do is to manipulate these kinds of systems pharmacologically. We're talking about drugs. What we would like to be able to do is to recapture in animals and humans the effects of dietary restriction, but without the dietary restriction. So there's a lot of interest in drugs that can extend lifespan. And one of the ones that's come to light recently, which is again part of this nutrient sensing network, is rapamycin, which of course inhibits the target of rapamycin, hence its name, TOR, which is a crucial nutrient, intracellular nutrient sensing kinase, which its pathway interacts at multiple points and feeds back on the insulin signaling pathway. They're really just one great big signaling network. And there was a lot of excitement a few years ago when it was found that feeding rapamycin to mice extends their lifespan. So you can see from the red arrow where the successful feeding started. They actually thought they were administering the drug before that, but it wasn't getting into the mouse. But they eventually formulated it correctly, and you can see the increase in lifespan. Those are actually the cumulated results of 3 independent studies in 3 different centres in the US. It's part of the National Institute of Aging Intervention Testing Programme, where each year they take on 3 or 4 drugs and test them in these 3 centres, and rapamycin came up in all of them. They also did males, and they saw an extension of lifespan, again, less than in females, but it was there. And of course, rapamycin's a licensed drug, which makes it of particular interest. Its on-license applications are for preventing restenosis after cardiac surgery, as an immunosuppressant in tissue transplant, and because of its growth inhibitory effect as an anti-cancer chemotherapeutic. But as you would expect for a drug that might be interfering in the actual aging process itself, turning out to have a much wider therapeutic range than anybody realised. So these are just a couple of studies showing this. The first one's actually a study of the consequences of inoculation against influenza in mice. So if you infect a young mouse with influenza virus, you can see the dark line going down, that's what happens to the survival of young mice without immunisation. So this is a very challenging virus. The horizontal line at the top is what happens in the young mouse if you do immunise it. So the immunisation is extremely effective. However, the dotted line, sadly, is what happens if you immunise an old mouse and then challenge it with the virus. It's almost as badly off as a young mouse that was never immunised. And what they found in this study was that if they pre-fed, or pre-treated, I should say, the old mice with rapamycin and then immunised them, they're actually part of that horizontal line at the top. There are two lines there. They could completely restore their response to immunisation. And they eventually figured out that it was an effect on the hematopoietic stem cells, which are, of course, important in mediating the response. And I think nobody had suspected this because rapamycin at much higher doses is used as an immunosuppressant. And it also turns out to be effective against these animal models of Alzheimer's disease. This is shown with this paper here that came out about 4 years ago now. They simply treated one of the Alzheimer's mouse models with rapamycin, and again effected quite a major rescue, leading to this really quite thoughtful review about rapamycin. Again, flies have proved very useful here because they also respond to rapamycin with an increase in lifespan. This is some work that was done by Ivana Bedov, who was a postdoc in the lab at the time. She actually now has her own lab at the UCL Cancer Institute. But she was able to go downstream fairly rapidly and work out which targets of TOR are important in the response. Found that the S6 kinase and the increase in autophagy that happens in response to treatment with rapamycin were both required for the increase in lifespan. But I think much to most people's surprise, Florey BP, which mediates the response in protein synthesis, was completely unimportant. Mutant flies that completely lack that protein respond normally to rapamycin. And interestingly, it's since turned out that the S6 kinase, again, this is a result of our Wellcome Consortium work in Dominic Wither's lab. If he knocks out the S6 kinase 1 in mice, he sees a very nice increase in lifespan. And I think as a result of that, there are quite a few clinical trials starting up in various places with lower doses of rapamycin than are normally used. This is just one that was highlighted in Science News at the end of last year, where they're looking at a group of older men, and they're looking at progress from mild cognitive impairment and also the effect on the mobility of the men in the trial. And this theme of a broader therapeutic range of existing drugs is becoming an increasingly frequent one. So metformin, I think, is a very interesting case. It's now the first line of defense against type 2 diabetes when it's diagnosed. But it wasn't always so. And at the time when metformin was used, but so were two other treatments, including direct treatment with insulin, because they kept such good clinical records in Tayside, they were able to compare other outcomes, other health outcomes than the diabetes in individuals who were either allocated to metformin or to the two other treatments. And very interestingly, they found that the incidence of various cancers was almost halved in the people who were treated with metformin. It really is a very big effect, and it's several different cancers, which I think is very interesting. And even the humble aspirin, I'm sure you've seen this in the news, it's turning out that users of long-term, low-dose aspirin are also protected against various cancers. So I think the way that people in this field are starting to think is that if you want to protect against the diseases of aging, then there is the prospect of protecting against the ageing process itself as the underlying risk factor. And to produce something of a parody, if we think about the major bad diseases that can happen with ageing, at the moment, the research is not entirely separate, but it is true that we all tend to work in different research institutes with the name of our disease as the headline for the work of the institute. And we go to different conferences and don't necessarily communicate very much with people working on other organs or problems. And that maps pretty well onto the different clinical specialities. But I think what we have with the animal work is proof of principle that we could be trying something else in parallel, and I think we should be, which is intervening in the underlying ageing process itself to try and ameliorate these conditions simultaneously. And there's growing interest in the health economics of this. This was an article that was produced quite recently, just looking at how much money would be saved if this approach was used and as effective as it might be at fairly modest assumptions. And I think increasingly people are thinking in these terms. I've mentioned that a lot of existing drugs could be repurposed, and that's true, but we would also very much like licensed drugs against some targets for which there aren't any, particularly the canonical insulin signaling pathway itself. And there, in the development of new drugs, I think there's going to be major challenges. These are just some of them. So, I mean, the goal is to keep people healthier as they age. Well, ageing isn't a recognised disease for a clinical trial. So there's a problem there straight away that you would have to specify a sub-aspect of disease as things stand. We also don't know, even from the animal models, because this information is so relatively new, how long and when we would actually have to treat. People are looking at timing. Does the animal have to take it from the beginning of adulthood? Is it good enough to start in middle age? And so on, but we don't know that. And obviously, if it's lifelong, it could be problematic. There's a huge amount of feedback in these pathways, so we'd almost certainly be looking at a combination of drugs to try and prevent the pathway fighting back if you inhibit a particular pathway. So long-term and perhaps more than one ingredient, major safety issues, clinical trials, the usual kinds of problems. So how much we're going to see the development of new drugs in this area, I'm not so sure, but we'll see. So, to summarise, I think what we've found over recent years is that the ageing process really is malleable to genes and diet and drugs, and the long-lived animals are healthier, and they're protected also against the pathology associated with specific ageing-related diseases. And I think we hope we have the prospect for a broad-spectrum preventative medicine for the diseases of ageing. And finally, it's a great pleasure to show you the people I work with. So I'm very, very lucky, actually, to have two labs. I have a fly lab here at UCL, where I'm involved in this wonderful Wellcome collaboration. And I also have a Max Planck lab, where we work mainly on mice. And this was the joint lab meeting of the two groups earlier this year. And these are the four people who you see here, particularly involved in the work I've talked about today with the Alzheimer's model in the fly. And finally, I'd like to thank you very much for your attention.